This is Twist. This Week in Science, episode number 890, recorded on Wednesday, August 31st, 2022. How to make an embryo. Wonder how we do that. Hey, I'm Dr. Kiki, and tonight on the show, we are going to fill your heads with heat, snot, and dolphins. But first, thanks to our amazing Patreon sponsors for their generous support of Twists. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Some scientists went walking out one dark and windy day upon a glacier tested in the scientific way. When all at once a mighty flow of melted ice they seen rolling down the jagged caps into the Arctic Sea. A bolt of fear went through them as the data would imply a warmer future coming, and it caused a mournful cry. Their faces got their eyes a blur, their studies soaked in sweat. They're trying hard to spread the word, but the world ain't heard 'em yet. Yippee-yi-oh, yippee-yi-yay. Greenhouse gases in the sky. The planet is on fire, the effects are going to last. Storms are getting fiercer and the seas are rising fast. People, change your ways today or forever you will try to chase that carbon capture across a greenhouse sky. Yippee-yi-oh, yippee-yi-yay. Greenhouse gases in the sky. This Week in Science on the way.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again with new video setups, new audio setups. We'll see how everything goes, but welcome. Thank you for joining us for this science, because that's what we're going to get into today. I have stories about embryos and rearranging evolution, a little bit of snot, and brains, because I love pattern finding brains. Brains and pains we'll talk about. Justin, what did you bring?
I have got zombie ice, 7,600-year-old tree, oxygen on Mars, and what type of break is best to take?
Any
break at all.
Take a break, everybody.
Blair,
Yes.
what's in the animal corner other than a little break from the rest of the science world?
I have beetle bacteria and dolphin alliances.
Ooh, alliances like dolphin friends. We'll figure that out. Alrighty. Well, it is time for us to dive into all of our science. And as we do, I want to get everyone to subscribe to This Week in Science. So if you have never subscribed to This Week in Science before, know that you can find us all places that podcasts are found. We release every single week. Our live shows are Wednesdays, 8 p.m. Pacific time on YouTube, Facebook, and Twitch. You can look for This Week in Science. Also, Twist Science. That's what you can look for on Instagram, Twitch, and Twitter. Our website is twist.org if all of this is too much. But if you're watching right now, or if you're subscribed and you're listening right now, don't forget to give us a like or a little recommendation to your friends. Let's get to the science. We have some great stories to jump into. So, first off the docket, how about making embryos from scratch? First you add a little, flour, couple of eggs.
Definitely eggs.
Milk. Yeah, well, actually, in this particular case, these researchers from University of Cambridge and a bunch of other universities working together, they didn't use eggs. They didn't use sperm. They used stem cells. And the stem cells were the very interesting thing about this particular effort at creating synthetic embryos is like at the beginning of the month, I had a story of a group out of Israel who had successfully synthetically created mouse embryos that lasted up to 8.5 days, which is the same amount of time as this particular technique, but they're using different techniques. And the Israeli group, they had been using pluripotent stem cells that could be reprogrammed and given a chemical bath that allowed them to start turning into the little embryonic cells to create a blastocyst. This team just published in Nature this week on their effort in which they have been finding just the right balance of different kinds of stem cells. So they're using trophoblast cells, they're using extra embryonic endoderm cells, and inducible extra embryonic endoderm cells. A lot of technical words, but really what they're doing is they're finding the right balance between the different types of cells that are going to go into creating the embryo itself, and then the other cells that go into creating the placenta and other supportive cells that actually add to the ability, the nurturing ability of the environment. So instead of creating a fake womb necessarily, they are creating this ball of cells that is self-supporting and self-sustaining and has all the things that it's going to need. They were able to show after 8.5 days of this in utero development of a mouse embryo model, they had head folds with a defined forebrain and midbrain regions, and they had a beating heart-like structure. They had a trunk comprising a neural tube and somites, which are like little what could go on to become body parts, tail bud containing neuromesodermal progenitors. Lots of interesting technical science words here that all come into basically creating a little natural, synthetic embryo that matched the development of a normal embryo.
So let me ask this.
Yeah.
Genetically, is this a clone of whatever they got the stem cells from? Is this like a mammal budding, basically?
Yeah, right. So because it's not sexual reproduction, it's not an egg and a sperm, and it's stem cells. This is creating. Yeah, it would be a clone. This is not sexual. This isn't sexual reproduction. Yeah. Mm-hmm. It's not recombination in that manner. Yeah, so that is interesting. So we're talking about it as, and they're not even talking about it in terms of cloning or reproduction so much as creating a model for studying embryonic development so we know how all these different parts work and how they develop, how the different types of cells work together so that eventually we can create systems where we can grow organs for adults. So that's the key idea that this particular research team is working from. The other aspect that they were quoted on several times is getting to the point of being able to understand what it is that allows an embryo to latch onto and the uterine lining so that there's actual the fertilization and all of the processes that take place that go into developing the placenta and all this stuff. There are a lot of problems that occur even before the embryo attaches to the uterine wall. And there are lots of miscarriages that happen even before that attachment takes place, in which women don't even know they've been pregnant, but that if genetically, if things go wrong, then it's going to keep a pregnancy from coming to term at all. Yeah. So babies, but not babies. These embryoids, as they're calling them. Embryoids.
Organ farm.
Yeah. Embryoids for organs.
Yeah.
Yeah. Yeah. So who knows?
Yeah, I love this idea, especially the idea that we could get it to the point of just creating a replacement organ in a dish, if you will.
Yeah.
Because as much as I would like to have new organs through science, I'm honestly a little bit freaked out if they had to make a whole Justin that they were just farming from. There's
just the liver. That's all we need.
Yeah, if they just made the liver, I wouldn't feel like, hey, that's me over there. I would feel like, hey, that's a liver I could use later. But if it's a whole me that they had to clone and keep living in the dish, oh gosh, then I start to have questions about whether or not I'm the right one. You know, test him. Maybe, like, what if they test him and it turns out he's a smarter, better singing version of me? What if he can hit his yippee-i-os and yippee-i-as right on the first go-around, you know? Then they might look at me and go, hey, we better keep him around because we might need his organs later for...
No, all your organs have been around. They don't want those.
It won't be...
Sometimes,
yeah, you're right.
But sometimes things stick around for a while when you don't really want it to. I mean, we like ice, right, Justin? But what is this zombie ice? Zombie ice sounds bad.
Well, you know, you think that zombie ice would be a bad thing because of the fact that they're calling it zombie. So and it is. No, that's right. That's probably why they use that term. The current models used by the IPCC Intergovernmental Panel on Climate Change predict sea level rise from Greenland's melting ice of 6 to 13 centimeters, or 2 to 5 inches, if you were so inclined. This is a prediction of sea level rise from the melting by the end of the century. So that means that most people alive today, with or without an organ farm, won't be around to see it happen, most likely. But a child born today, pretty good shot at being around for the prediction, to see if those prediction models are accurate. However, as often seems to be the refrain around here, the IPCC models have tended towards gross underestimation of the severity of global warming's pace. Published in the journal Nature Climate Change, glaciologists at the Geological Survey of Denmark and Greenland have been studying ice loss in Greenland and have released a study that pretty much doubles the rate of ice loss estimated by the IPCC by the end of the century as the low end of their prediction. And actually, it doubles the high end of the IPCC's prediction as their low end. And it's all because of zombie ice.
Yeah, what's
ice? Ice. So ice caps. You know, you get more ice in the winter, it builds up, and then comes the summer, it kind of shrinks a little bit, and then it fluctuates. It goes back and forth. And most ice can grow. It can have it can have snow or other water melt onto it and then freeze again. Zombie ice is ice that is no longer in a position where it is no longer able to accumulate new ice and get bigger. These areas of the glacier are in the process of becoming seawater, and their direction throughout future time is pretty much set that way. And it's about 3% of the Greenlandic ice cover now, which is not going to recover by the end of the century because it is zombie ice.
So it's called that because it's basically already dead. Like, its fate is sealed.
Yes, exactly.
Okay.
Yeah. So even if we follow the Paris climate guidelines, even if we actually stopped all greenhouse gas emissions today, they found that Greenland's ice loss under current temperatures will raise global sea level by at least 27.4 centimeters, 10.8 inches if you are so inclined. That is the low end. Actually, it's not actually the low end. That's the baseline commitment. That's the already made the down payment. That's what we've committed to in sea level rise from Greenland's ice sheet, even if nothing else changes.
Mm-hmm.
The same study said it could reach as much as 78 centimeters or 30 inches of sea level rise from the zombie ice. But it doesn't mean that the sea will rise by this much. That's just the contribution of one major source, not including anything going on elsewhere in the Arctic or in Antarctic regions or anything like that. That's just the Greenlandic ice sheet.
Yes, all our oceans are connected.
Yeah. And there is, and it's, of course, also, the lead author on this is a gentleman, Jason Box, who was originally from, I want to say, University of Boulder, something like that. But went to a talk he gave here in Denmark, where he was doing a really nice job of explaining why sea level rise also isn't going to be universal everywhere. As the Greenlandic ice cap melts off, a tremendous amount of pressure is going to be relieved from that northern Arctic region, which will actually allow land levels to kind of rise a little bit because of all the weight and pressure that's no longer pushing down on them. Also, there was a wonderful graph that kind of showed relative predicted sea changes. The North Arctic actually didn't get that much. The coast of California, actually, for some reason, was also not as bad as other areas. But most of the world around the equator was the most dramatically affected, which is very unfair because around the equator tends to be regions that have contributed the least to greenhouse gas emissions. We'll be seeing the majority of the sea level rise.
Well, isn't a lot of
zombie ice? IPCC reports again underestimated, and again, this is all the minimum commitment is if nothing changes, and we had stopped everything today. So
nothing changes. It's still going to be a big deal. And the equator is very likely because of the squishing, because we're rotating and we have a moon that's going around also. So there's a bulge and the squishing. We're an oblate, spheroid, very oblate because of that inertia that we have.
You're so oblate.
You're so oblate. Who's oblate? My child's going to be oblate. Never mind that.
Grouchy Gamer says, I'm wondering what the end result will be. A lot of major water sources seem to be drying up, but sea level rise is also predicted. It's all got to balance out at some point. So one of the things that has been a long time prediction of a lot of the global warming scenarios is with all that evaporation of the poles, you are going to also have a situation where there is more atmospheric moisture. So while you will have droughts and strange weather patterns that don't match the biomes that are currently there, you are likely also to have more severe storms or areas that get appreciably larger amounts of water. The problem with saying like, oh, well, we're going to get more fresh water from the sky is we're not prepared for 10-20% increase in one area over one season. This is like, look at the current floodings that they're all over the news.
Well, and a lot of the planet is covered in salt water, and so that rain will fall into salt water and not help us at all.
Yes,
so
but I mean,
that's part of it.
The parts that do fall over the land should be increased by a percentage as well. So that's
right, but then
that
drought
part,
conditions make it so that they slide right off and don't go into the water table, and then they are lost. Also,
or just you have a river that isn't ready for what is that? What's the term? An atmospheric river to be added to it. Like,
Yeah.
What was it? What's the part of the states that's being flooded right now? Is it Mississippi? Where they're saying it's indefinitely they're not going to have drinking water. This is, water everywhere, not a drop to drink. Because when you have flooding, you have overturning of banks. That water that's going through cities, where you have wastewater treatment plants that have overflowed, or water treatment plants for water municipal districts that are inoperable. Having clean drinking water is more complicated than just having more water.
Yeah, and it'll all be affected even more as sea levels rise, because as sea levels rise, they push inland, and so then you have those rivers and all the rivers go higher up inland, and then the flooding is worse, and the groundwater table can be inundated by salt water. There's all sorts of issues.
Yes, the saltwater intrusion up the deltas.
Yeah.
Yep, absolutely.
Yet to come.
Not great.
Speaking of not great, Blair, you have
that is quite an
important story, but not great. Let us know.
So sometimes science can help us understand common public health and kind of public crises in our populace. And so this is a study out of Florida Atlantic University looking specifically at teen suicide rates, and that linked to something called digital self-harm. So I did not know about this, which is part of why I wanted to talk about this. We're a show on the internet, but I suppose I am no longer in the know with what teens are up to online, and so this is really helpful information, actually. So digital self-harm is the online posting, sending, or sharing of hurtful content about yourself anonymously. Yes. And so if you look at traditional forms of self-harm, cutting, burning, hitting yourself, there's a lot of research that links that to suicidal ideation and attempts. And so they wanted to see through this research if youth who post cruel, embarrassing, or threatening content about themselves while their peers assume a third party is the culprit, if they do so for similar dysphoric reasons that could lead to similar suicidal ideation or attempts. And so they did, in fact, find that. Findings serve as a warning sign that youth who engage in digital self-harm may also be engaging in or at risk for suicidal thoughts and behaviors. So looking at their study, they showed that approximately 9% of adolescents reported that they had anonymously posted something online about themselves that was mean, while about 5% said that they had anonymously cyberbullied themselves. With regard to suicidality, about 8% of adolescents reported they had thought seriously about attempting suicide in the past year, while 5.3% said they had attempted suicide during that time period. The finding within that is that those engaged in digital self-harm were between 5 and 7 times more likely to have considered suicide, and between 9 and 15 times more likely to have attempted to end their life. There was no significant difference across gender or race, but, as has been the case in previous studies looking at this sort of thing, non-heterosexual students were significantly more likely than heterosexual students to have seriously thought about attempting suicide. 24.5% versus 7%-ish. So it's a huge difference. And to have attempted suicide, 10% versus 4.9%. Samir Hinduja, PhD, co-author, and professor at the School of Criminology and Criminal Justice within the College of Social Work and Criminal Justice. He's also the co-director of the Cyberbullying Research Center and a faculty associate at the Berkman Klein Center at Harvard University. lots of credentials from this individual, says, quote, "We have identified a strong association between digital self-harm and suicidality. As such, it's imperative for health professionals to screen for digital self-harm to address underlying mental health problems among youth that may occur prior to or alongside suicidality. Moreover, parents and caregivers must convey to children that they are available to dialogue, support, and assist with the root issues that they may eventually manifest as digital self-harm. Educators and other stakeholders must better prioritize the emotional and psychological needs of youth with opportunities for open dialogue, skill building, and the provision of interactive and easily accessible online and offline support resources to ensure that those at risk know that other people care about them." So this is just a reminder for those of us that, I thought I had a good idea what was going on online, but I didn't even know about this thing. So, especially for those people out there listening who work with teens or have teens at home or are close with teens, keep an eye out for this kind of stuff on their social media presences. If they're being cyberbullied by another person, that's also not good for their mental health. But
Right.
if there's an inkling that it might be digital self-harm, making sure that those individuals know that there are resources out there for them and that there are people out there who care about them is really important. I'll also throw out there, I did a youth mental health first aid course a few years ago, and it was amazing. I think a lot of them are free. The ones that aren't are pretty reasonably priced, but there's a lot of resources out there to be able to serve as a lifeline to any teen in need, especially those contemplating suicide. So just wanted to throw that out there. There's also lots of self-help lines, texts, calls, all that good stuff. There's resources, but this is something extra to look at that we know about thanks to this research.
I mean, how do you look out for something that's not a physical manifestation? So the other behaviors of self-harm, there's usually, you can find physical signs at some point. They're apparent, but on social media, how do you track that, and how without being invasive, of their social media privacy? And this is a new area, and there are so many questions. And how do you watch for this, and how do you?
Yes,
Well,
and I will also
how do
say,
we talk about it? I mean,
other
you've done a good job,
teens,
but,
right?
Yeah.
The people who know people online who are being cyberbullied or who are suspected of maybe doing digital self-harm. It doesn't all have to come from adults coming out going, "Hey, I'm here for you, buddy." Like, peer-to-peer support is huge with youth. So that's also something to keep in mind. If you know a youth in your life that has the resources to become a peer-to-peer mentor, that is also a great thing to get them involved with. So yeah, I wanted to call attention to this because I had no idea about it.
Thank you.
And I mean,
No idea either.
When we're talking about physical manifestations of these things, it's actually an additional one that we normally don't have. I would think, because unless this is there's a journaling self-loathing habit, which I'm sure that's a thing when people are keeping a diary or something like that to write down negative self-thoughts and self-impressions, because that's what a lot of these things are, is they're negative feedback loops in the mind that become these other entities that are attacking the self. That we have access to social media, and that it is now what would have been maybe a diary or journaling or that sort of a thing where somebody's jotting down those self-harm things. It actually gives us another physical manifestation in the world to try to catch these things. So you're right. Very important that we are aware that exists, and that's a thing to look out for.
I found the line that I found out about in my youth mental health first aid class. It is 741-741 in the United States. You can text 24/7 and someone will respond. There's also the new 988 number that you can call, and I think they receive texts as well, and that is also in the United States.
Thank you for those resources. Mm-hmm. Thank you, Blair. And moving past that serious note that we should all keep an ear and eye out for, I'm gonna talk a little bit about rearranging evolution. How do new species come about, right? We've talked about Darwinian evolution, and through the years, once upon a time, I said, bah humbug to Lamarckian ideas, and now we've got epigenetics in there
Yes.
doing doing double time.
Sorry, Lamarck.
Sorry.
I never gave up on you, Lamarck.
You never
Just
did,
for the
Justin.
record, I was with you all the way.
No, but in this, there's this idea also of just kind of the long march of evolution, tiny changes through mutations, eventually leading to ta-da, a new species. Right? There's more to it than that. There's also a process called chromosomal rearrangement, and chromosomal rearrangement takes place different time-scales for different organisms. And mice, with their faster breeding times, they have a faster chromosomal rearrangement time than, say, humans with our much longer reproductive times. And researchers wanted to dig to the bottom of how rearranging chromosomes, moving around which genes are on which chromosomes and how many chromosomes are involved in the process of making a new organism. Also, which genes are imprinted or basically marked as the ones that are going to go into creating the organism in the next generation. How all these things fit together and to create these evolutionary paths. Specifically, they want to know, how did humans get to be humans? How did we get step by step the different species that branched off until humanity branched off and became Homo sapiens? So we have chromosomes that are, our 46 chromosomes, right? And we have a different type of chromosomal separation organization than, say, gorillas. So there are chromosomes that stay separate in gorillas that get fused in the human genome, or that were fused in the human genome. So the researchers demonstrated by smushing a whole bunch of chromosomes together, basically fast forwarding the evolution timescale for mice. They created fusions of chromosomes 4 and 5 in mice, and they did some other also smushed together chromosomes 1 and 2 into 2 different orientations. And so they gave a bunch of different arrangements of chromosomes through their genetic editing. And in doing it, they were able to create new mice that reproduced a little bit more slowly. They didn't, the fusions of chromosomes 1 and 2 didn't produce any mice offspring, but others, the 4 and 5s, their code got passed on to mouse offspring, even though they had slower breeding. And so the researchers say they experimentally demonstrated that the chromosomal rearrangement event is the driving force behind species evolution and important for reproductive isolation, providing a potential route for large-scale engineering of DNA in animals.
So they've created new species of mice then?
Kind of, yeah. They're basically creating mice that won't be able to breed with other mice anymore because they're chromosomally going to be different.
Wow.
So yeah, in a sense.
That's wild.
Yeah, it's a fascinating area of study, and they say their research article is in Science: A Sustainable Mouse Karyotype Created by Programmed Chromosome Fusion. So there's this kind of fusion and work has only really been done in yeast previously. So to have it work in haploid embryonic cells working with mice, it's a pretty interesting step. And in this chromosome ligation, where they cut chromosomes and created these new karyotypes, there were different challenges and new challenges, but they were successful. Yeah. So yeah, chromosome level engineering toward the creation of new species. Oh, humanity, what can we do next?
Cool.
Where will we take it? Oh, but I mean, will we have much time to do that? I don't know, Justin. Do we have a lot of time to dig around with our dig around in our chromosomes and make new species?
No.
Okay.
No, the world is ending fast. Actually, this is my next story is from Siberia, which is recording the warmest summer of the past 7,000 years. So for several thousand years, the temperature of the region was actually cooling a little bit, generally cooling a little over the years. Then, for no known reason, there was an abrupt change over the past 100 years or so with rapidly rising temperatures. Research into the region has been published this week in Nature Communications. The current warming is well recorded in recent times, but finding the history of the climate in the region fell to an amazing collection of wood fossils collected over the past 40 years by a Russian researcher, Stefan Shayatov, with colleagues from the Institute of Plant and Animal Ecology, a branch of the Russian Academy of Sciences, began collecting these subfossil wood specimens about 40 years ago. Currently has a collection of more than 5,000 samples. They spent a lot of time measuring tree ring widths by dating and overlapping tree rings between different fossil trees. They form a sort of virtual tree that has been growing for over 7,600 years. A tree ring record that can provide firsthand data on what climate activity was doing throughout all those millennia. Not only do they show that the temperatures reached unprecedented warm levels, but also the rate of temperature increase is fastest over the past 150 years. That the rate has been increasing as well. Last century was characterized by a total lack of cold extremes. So nothing in the current record is showing extreme cold temperatures in Siberia, of all places, contrasted by the occurrence of 27 warm years, 19 of which have fallen in just the last 40. So extreme warm years is now the norm. Research on tree ring-based reconstruction will continue, and they're trying to push it back. They think they can push it back another 2,000 years or so from new fossils and that they've been collecting. But I just love the, this is always one of the problems is like, how do you look back? What are this, how do you know how do we know what we know about past climate? This is one of the ways that's a new one that's been added to the list of things that show climate over millennia. Is all these tree rings overlapping and all these different fossils. Very clever way
and
of
I'm
going
sure that we'll get more fossils as
about it.
as the ice recedes, as areas melt a bit, and fossils are uncovered as they come out of the ice. Even.
Yeah, and you've got to be quick because there's a lot of territory and a lot of ice that's melting all at once. So there's going to be a need for people who with good mud shoes to be walking around looking for this stuff, artifacts, archaeological artifacts, paleontological artifacts, all sorts of interesting stuff is going to be discovered as the ice melts. But we're going to have to be quick if we want to recover it. There's another story I didn't bring, also that went back and looked at some of the climate estimates for temperatures going back like 65 million years when the carbons were really high.
Mm-hmm. And it was hotter, right?
Yeah, it turns out it was a good perhaps 25% hotter than the other estimates. When they looked at their data, it was much warmer than the past. And I think they were looking at seashells or calcium from sea creatures in the fossil record, something of this nature. So
Great.
So that's 65 million years ago. And that's a tough one because that's going way, 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 way back. But this is this one here with the overlap of the tree rings. And I've also, I don't think
it's just hot. It's too hot. It's too darn hot. The rent's too darn high. The temperatures are too darn hot. It's getting too darn hot and it's our fault. And, so let's do something. Let's not doom and gloom it. We have to start talking about more solutions. Energy production. Yes, we can do it.
Yeah. Okay. Well, I was just going to say, I really love, because this researcher who started collecting and measuring and finding overlaps wasn't doing this as a climate change study. They were just really into tree rings.
Mm-hmm.
They were really into studying trees.
Sure.
Yeah.
They're like dendrochronology. It's awesome.
Nobody even knew what they were. They're like, oh, I think he studies dandruff. I'm not really sure what this guy's doing. And he got enough other people interested, got a little bit of funding. He's been collecting these tree rings that now shows a 7,600 year history of climate and rainfall and all sorts of other things for the region. So I think I like that.
Yeah, turns out this is where all those conspiracy theories about research scientists in it for the money. Yeah, I mean, this researcher in it for the
making the dough hand over fist from these fossilized tree rings.
Yeah, make great footstools. Yes. What's been around for a long time, probably definitely longer than trees and the tree rings.
What's that?
Snot.
It's not what?
Snot has been around for a long time. Mucus, the slimy, ewy, gooey, wonderful stuff that comes out of our heads when we have colds or during allergy season. Mucus that helps many organisms eat, to adhere to things, to catch their prey, to have sex. Mucus, it's important for so many things in nature. It's been around for years, but where the heck did it come from? How did we get slime? And it's not Nickelodeon's fault. Why did we get I know. Why did we get so slimed? Why did all mammals and animals end up with so many what are called mucins?
Lubricants, right? It's just like any machine, you need a lubricant, right?
Yeah.
Learned from nature.
Exactly. But researchers at the University of Buffalo have just published their study into the evolutionary history of mucus, trying to figure out in mammals where it's come from. And in their investigation, they looked at 49 mammal species and identified 15 times where new mucins evolved from just adding a protein into a mucin. So you had a mucin, which is a protein type, and there was what they call mucinization event. So at mucinization, there was a protein. It wasn't a mucin. Then they got this new part that had amino acids, with a bunch of sugars on it. The amino acids with the bunch of sugars got stacked onto the protein, and then another one, and another one, and another one, and eventually they have all these amino acids with these sugar brushes. And that's what makes a mucin. And so it's this mucinization can basically grab a protein that wasn't a mucus protein before. The process turns it into a mucus protein, and then you have slime. And so these researchers are saying that this, what's called doubling regions, the repeats, the genetic repeats, over time, these regions get duplicated, and these mucins then become evolved and genetically useful, and the proteins are useful for their mucus factors. And it's a very interesting study. Can we now make mucins? Could I take a protein and turn it into a mucin in this way? They did an assay looking for the potential for different proteins to become mucins and identified a whole bunch of proteins that could turn into mucus proteins, possibly. They identified commonalities between, say, mouse mucins and human mucins. Mucin everywhere.
But not a drop to sneeze?
No snot. I can't take it. But snot to drink. No, I don't know.
Yes.
Anyway,
Mucinization. I'm gonna call it a mucinization event next time I sneeze. Excuse me, I'm experiencing a mucinization event.
Oh, they also were calling this the mucinome. They were like the genome of mucin. So they've now also added the ome suffix to mucin. So they have a mucinome that they're investigating.
Cool.
Yes.
So much of life couldn't exist without mucus. It's a pretty important event.
Mucinization. It happens over and over again. This means there's an adaptive pressure, makes it beneficial to continue the process, the repeats, the genetic repeats, something pushes it down that pathway to the organism says needs to be snot.
Perfect.
It is perfect. It is. And this is This Week in Science. Thank you so much for joining us for this episode, bringing you all the science that's fit to sneeze at, to talk about, to discuss. We're so glad that you are here today. Oh, and at this point in the show, I would like to invite you to invite a friend of yours to join us for Twists. Bring them to Twists next week. Get them to subscribe. Tell them how much you love Twists and share it today. And right now, you know what we have coming up right now on This Week in Science?
What's next?
Blair's Animal Corner.
It's Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
So first I want to talk about beetles and their bacteria. So beetles, just like us, beetles are just like us. They have some beneficial bacteria that are really important to their life cycle. Specifically, the females, they have bacteria that are beneficial microbial partners. And they start out on the eggs. When females lay eggs, the bacteria are squeezed out of glands next to the oviduct and deposited on the egg surface. Antibiotics produced by the bacteria protect the eggs, the larva, and eventually the pupa of the beetles against fungi. But where this gets interesting is the fact that, when a insect, I will be biologically correct. A bug is a very specific type of insect. When an insect goes through metamorphosis, it basically turns into a mushy soup in there. I'm being scientific. And so, how exactly does a bacteria on the surface of a pupa end up in a gland next to the oviduct of an adult when they're in this soupy mess when they go through metamorphosis?
No idea.
Yes. Great question. I'm so glad you asked. Yeah. So the kind of the added confusion to this is that the largest component of this symbiotic mix is a strain of bacteria, a strain of Burkholderia bacteria called LVSTB, which has lost the genes and cellular structures for motility. So it can't move. And can't survive for long outside of the beetle's body. So this is quite a quandary. So it turns out that these beetles actually have a specialized pocket on their back The bacteria hide out in there during metamorphosis. And then when they emerge as adults, the bacteria shuffle back out of these pockets backwards and then into their genital area where they can be held in those glands by the oviducts. So they have modified.
But they don't shuffle because they lack motility, so they're transported somehow.
Indeed. So these Lagria beetles, they're in the genus Lagria. They are able to keep the protective symbionts and facilitate the relocation to adult organs through these modified pockets. Males have the pockets, but they are pretty much only vestigial on males. They contain few to no symbionts because the males don't lay eggs, so they don't need them. They are there to be put onto the eggs as they are laid. Yeah, so they hang out in these 2 or 3 lobe pockets on the back of the thorax, where not only are they stored, but they are nourished. And then what they think happens is that when they wriggle out of their pupa casing, they think it gets redistributed and spread backwards towards their reproductive tract. The way that they figured this out, they scattered some polystyrene fluorescent beads across pupae, and then after emergence, the majority of these beads ended up on the tip of the abdomen. So that makes it look like they are forcibly being pushed backwards just by the emergence process.
Wow.
Yeah, the way that they then get inside the beetle and colonize the accessory glands, they don't know that yet. So what they want to figure that out. They want to figure out which host and symbiont factors regulate the establishment. For example, does the host select for specific symbionts? Like, are there other bacteria? And they're like, get out of here. Only these bacteria in these pockets. And through which mechanisms can they colonize specific organs? Because if by force you're pushing it backwards, that's great. If you get it to the genital opening, that's great. How does it then get inside and into these glands? That is really the last question in this story. But in the meantime, very cool that these beetles have adapted an ability to harbor bacteria during the strange and topsy-turvy reality that is metamorphosis.
That's amazing. We'll put you in a little pocket and we'll keep you there somehow. Not really sure, but it'll happen.
Yeah, that's pretty mind blowing. Really, this is a very I'm immediately extrapolating to if humans had to do some sort of thing, right? and it would be up to maybe your midwife or your nurse when you're giving birth or when you're born. There would be third parties involved for humans.
So there's something I can give you a similarity in a mammal.
Okay.
Which is koalas.
Mm-hmm.
They have the beneficial bacteria in their gut that allows them to eat eucalyptus.
Yep.
The babies eat their mother's poop in order to seed their own microbiome. Right. So that's all this is, right? Is a seeding mechanism.
Yeah.
So, but that is a very deliberate thing that is happening, and this is somehow this sessile bacteria is like migrating to these pockets and then migrating down. So it's a little bit more strange than that, but it's similar. It's basically you're seeding a microbiome from one generation to the next for a protective capacity, I guess.
But you need pockets.
Yeah, you gotta have pockets. This is the very first back pockets like this in larvae that they have recorded in insects. And I imagine once they start looking, likely there will be others.
Where one beetle has pockets, others will want them as well.
Yes.
Because pockets are amazing.
Oh my God, I love pockets.
We all love pockets.
Speaking of things that you only see when you look for them, My next study is from an international team of researchers led by the University of Bristol, and they found cooperative relationships in dolphins that are similar to alliance networks in humans. This is the largest one they have ever seen in animals outside of humans. So you can already guess how I'm going to talk about this story based on how I started it. But
Yes.
let me give you the facts first. Colleagues from University of Zurich, University of Massachusetts, and the aforementioned University of Bristol analyzed the association and courtship data to model the structure of alliances between 121 adult male Indo-Pacific bottlenose dolphins at Shark Bay in Western Australia. They found that male dolphins in Shark Bay form first-order alliances of 2 to 3 males to cooperatively pursue courtships with individual females. Second-order alliances of 4 to 14 unrelated males competed with other alliances over access to female dolphins. And third-order alliances also occurred between cooperating second-order alliances. So, we already know dolphins are really social. We know they can recognize each other in multiple ways. We know that they can solve pretty complex logic questions. So, all of that information together, it should not be too surprising that they have figured out how to work together. So that's what this is the largest one of these kind of quote-unquote alliances that we have seen outside humans. Co-lead author Dr. Stephanie King, who's an associate professor from Bristol School of Biological Sciences, says, quote, "Cooperation between allies is widespread in human societies and one of the hallmarks of our success. Our capacity to build strategic cooperative relationships at multiple social levels, such as trade or military alliances, both nationally and internationally, was once thought unique to our species." You can guess where this is going. Not only have we shown that male bottlenose dolphins form the largest known multi-level alliance networks outside humans, but that cooperative relationships between groups, rather than simply alliance size, allows males to spend more time with females, thereby increasing their reproductive success. So this is more than just large alliances. These are multi-level social organizations for mutual benefit. The duration over which they consorted females is dependent upon being well connected with third-order allies. So social ties between alliances had long-term benefits for courtship. So this also challenges some earlier expectations. One of the reasons that alliances are seen in humans, and scientists have previously thought that it was unique to us, at least in primates, is dependent on 2 features that distinguish humans from common ancestors with chimps, which includes the evolution of pair bonds and parental care by males. But these results show that intergroup alliances can emerge without those features. They have a social and mating system that is more chimpanzee-like. So they associated these kind of relationships that maybe are not relevant. All that to say, I also wanted to throw out that this all came from an anthropology department. And most of the time, anthropology departments research humans and primates. And that's usually where that ends. But an anthropology department who focuses on things like this, social dynamics, taking time to look at non-primate animals is really important because you come across things like this. And that's where I was kind of getting at the beginning of the story is like, when we look for things, we often find them. And so we understand how social interactions work in a lot of primates, but especially apes. So it's very easy to analyze that, categorize that, see what's happening. When you get somebody in the mix who understands marine mammals and specifically dolphins, then you can start to look at that. If you look at other animals that have less, like, quote unquote, human-like interactions, you might find similar alliances with beetles, with anthills who could ally and help each other out, right? You could find this in other areas in the animal kingdom if you know how to look.
Wait a sec. Are you saying that anthropologists were anthropomorphizing the dolphins?
Oh, what an interesting thought. Yes, I do believe that is the situation.
Okay. Well, I think that's their job.
Yes, certainly. Yes, that's the idea. Their whole job is to draw parallels between humans and animals and social interaction and all these sorts of things. So, absolutely. this shows that dolphin societies, as well as potentially those of other animals not yet to be looked at, could be valuable model systems for understanding human social and cognitive evolution, which is exactly what you're talking about, Justin. That's what they do. That's their bread and butter, is taking lessons learned from animals and applying it to human animals. And so I just thought this was a really interesting case of an anthropology department looking at dolphins. So cool. But also challenging that expectations of what kind of advanced social interaction looks like. And so there are alliances happening with dolphins. We just didn't know how to look for them before. And everybody, take a look. Look elsewhere. I bet you'll find more.
All those dolphin youth group football matches out in the ocean. Yeah, they serve a purpose.
Yeah, absolutely. And then they all team up against the regional champs.
That's right.
Alliances.
It's an interesting thing that anthropologists do, I think, in the study of humans, because so much of what we believe to be the reasons for our great intelligence, or the accomplishments of our great intelligence, or all of these sorts of things. We invented fire and started using fire, and that's a current modern human thing, except actually perhaps the human ancestors millions of years before that were already done. Oh, well, it's our social structure and our interreliance on each other that led it. Well, actually, dolphins and maybe even beetles are doing that too. So maybe that's so anthropologists are really, in a way, cutting away all the preconceptions we have about what makes humans special. And at the end of the day, we're just a hairless ape.
Well, and I think it's a good reminder that so much of what makes us special is based on evolutionary pressure that happened to us. It's not things that, like, there was a spark and we were different from all the other animals. There were social pressures that pushed us into these situations. We were extremely far reached. We had the braided stream. We had all these different hominids in different areas who had to work together or the opposite.
Die.
Right, exactly. So there were pressures causing these things to change that make us unique. And those same pressures can exist in other areas of the animal kingdom.
Indeed, they would. Yeah.
Still one of my favorite pressures for the human brain development comes about as having started as we now have become to realize, actually pretty much started as bipedal. But we had these big fleshy feet that were meant for being in trees a lot of the time. And the act of walking and then running created on these fleshy, blood-filled feet, created sort of a micro-pumping of blood that just kept forcing a little extra blood to the brain every step we took. And it's like this, like, nobody's like, all of these other things, this is why people are special and intelligent. And it could just be that we had fleshy, blood-filled feet that we ran around on that kept forcing more blood into the brain. And over, millions of years of walking and running around, it's like a brain's like, all right, you keep giving me blood, I'm going to make some more capacity to move this thing through here. Like, you solve this bottleneck problem of all this blood. And it led to greater intelligence. It's an accident.
Step you take will improve your brain. Your fleshy feet. Pump your blood. Okay, this is This Week in Science. Thank you, Blair, for an amazing animal corner. I'm hiding back here. No, I'm not hiding. I am jumping out, asking you, if you love Twist, to head over to twist.org and click on our Patreon link, please. If you enjoy our program and our conversations about science, please become a supporter of Twists and help us continue to bring you Twists every single week. Over on Patreon, you can choose your level of support, $10 and more per month, and we will thank you by name at the end of the show. Can't wait to include your name. Thank you all for your support. We cannot do it without you. Hey, Justin, What stories do you have?
I've got this is a meta study, one of those where they looked at other studies. This is a review of 22 previously published studies. It suggests that taking micro breaks is generally associated with reduced fatigue, increased vigor, and better mental health.
Oh, wait. Yep. Nope. Gotta go. See you, Blair.
And other studies show that if you work nonstop, you won't be rewarded at all.
I feel much better.
That was a little break.
I
Just
feel
a second there.
Much better having taken that micro break. So I love. Okay, so first of all, what really caught my eye in this study was one of the opening lines, which says, "Concerns are rising over the heavy workloads and long shifts faced by many employees currently in the workforce." And I immediately was like, "Wait, what non-American location was this study conducted? Caring about workers?" And so it turns out it's the West University of Timișoara, Romania. So it says, an increasing number of studies, not covered by American corporate media, are exploring various aspects of employee health. To improve understanding, this is Patricia Albulesco and colleagues at West University conducted this meta-analysis of the 22 studies from 19 different manuscripts published within the last 30 years, all of which examined the potential benefits of taking micro breaks from assigned tasks. Tasks varied between experiments and work, and included work simulations, real work-related tasks, non-work-related tasks, cognitive tests. Types of breaks varied, as well as times including physical breaks, relaxing breaks, more engaging activities such as watching videos. So statistical analysis of the combined studies revealed an overall association between micro breaks, which I think they've got identified as breaks under 10 minutes.
So that's just a trip to the bathroom, is what that sounds like, right?
And maybe
I take micro breaks all the time.
Maybe that's your micro break. Yeah.
I'm well hydrated.
But I mean, also, isn't this historically? There's the smoke break, there's the coffee break, there's the anything. There have been historically all sorts of breaks that people have worked into their day to try and have a break. And Blair, we were talking before the show, and you mentioned how you it wasn't accepted that you just took a break, and so you started drinking coffee so that you could say you're taking a coffee
drinking
break.
Coffee.
Then I started sipping coffee, and now I'm addicted to coffee.
Good for you.
I got my breaks.
So they found there was an association between taking those micro breaks and higher levels of vigor, lower fatigue in the participants, and it suggests that micro breaks may contribute to a person's overall well-being. No overall association was found between micro breaks and better performance on tasks, however. So your boss still might not let you take those micro breaks because you're not helping the bottom line when you do, just your own mental health.
Just your own mental health, right? Okay.
So this is, however, researchers did find that longer breaks tended to be linked with better performance, especially for creative tasks or clerical duties. So whether you're doing something creative or perhaps rather methodical, longer breaks did lead to better performance than a micro break. But it was less so for more cognitively demanding tasks.
That's interesting. So maybe if you're in flow, in a flow state, then that's different. Because sometimes you get into that mode of, not just creativity, but thinking about something, writing, like doing hard cognitive work, but you get in this flow of putting it down, and taking a break can break that flow.
Right. I'm also thinking about, I was working from home today. I'm very lucky to be able to work 1 to 2 days a week from home currently. And I have noticed that when I am physically at work, I'm definitely taking these micro breaks. I'm not doing it intentionally, but like someone will stop by the office and say hi. I'll go walk down the hall to the restroom and stop by somewhere where else and have a hallway conversation, or I'll walk around the building, or like I'll go do another task in another room. I'll just kind of like break up my day. But I find that when I work from home, not only am I very, very bad at taking breaks when I am at home, because I am at home, I'm in a comfortable space, no one's interrupting me. But I also notice that the last hour and a half of my workday, my brain is fried. And so I do think this study is also important, recognizing how much working from home is currently going on, because it is harder to kind of make that clear differentiation between kind of work and home, and how you have like these routines built into your day when you're at the office that might not exist at home. It's a different kind of social emotional space.
Yeah. But this does also support some of those beyond just social emotional stuff. It supports also things like the Pomodoro timer and other techniques that people use to add to work for a chunk, take a small break for a chunk, work for a chunk, take a small break for a chunk, and kind of enforce those breaks to keep your mental clarity and to keep yourself going. I don't know. So this interesting support.
Yeah, a little bit of the differences that they're also pointing out here is says here there's 3 attention networks. There's alerting, there's guidance, and there's executive control. That they kind of reviewed in doing this. And it seems like the short breaks are really good for alerting or attention. We're being able to refocus attention. You take a short break, you come back to your task, your attention level is better. But if it's but for the guidance and executive control, a heavily cognitively involved thing, the longer break is better. It was the one that actually boosted performance. So I guess you can tailor. Not all breaks are the same, so you might need to tailor the type of break you are taking to the sort of activity you're doing. And that might be further research in the future will tell us exactly what sort of break we should be doing based on the activity. Whatever. And then my last story is kind of a big deal. We are making oxygen on another planet.
Are we now? I knew we had done a little bit. We're doing a lot now?
Yeah, the MIT led, so we've been doing this for a while, actually. But they just published the study on the MOXIE, or MIT led Mars Oxygen In Situ Resource Utilization Experiment, aka MOXIE, has been successfully making oxygen on planet Mars since February of 2021, when they first got the touchdown on the Martian surface. So in the study published in the journal Science Advances, they say MOXIE was able to produce oxygen on 7 experimental runs in a variety of atmospheric conditions, including during the day and night and through different Martian seasons. In each run, the instrument reached its target of producing 6 grams of oxygen per hour, which is about the rate, they say here, of a modest tree back on Earth. So MOXIE's doing the job of one little tree on Mars right now. MOXIE works by capturing Martian atmosphere in its HEPA filters, compresses the atmosphere via a scroll pump, heats it to 800 degrees Celsius. Oh boy, that's hot. Sends it through a solid oxide electrolysis assembly where CO2 flows over a nickel-based catalyzed cathode and decomposes into oxygen ions and carbon. So the researchers have been running a number of these experiments because the Martian temperatures and atmospheric density fluctuates much more intensely than it does down here on the planet Earth. So you can have somewhere in here it says that the atmosphere's density can double at times, which is not a thing that we normally go through here on Earth. So, here we go. This is Hoffman. Jeffrey Hoffman is professor of the MIT's Department of Aeronautics and Astronautics. The atmosphere of Mars is far more variable than Earth. The density of the air can vary by a factor of 2 through the year. The temperatures can vary by 100 degrees. One objective is to show we can run in all seasons. And so far, MOXIE has shown that it can make oxygen at almost any time of the Martian day or year. And of course, then what's the next step?
More MOXIEs.
Send more MOXIEs.
Yes.
Yeah. So, and this is in situ. So this is not just releasing oxygen that it brought with it. This is actually converting the Martian atmosphere itself into that oxygen, which will make it one less thing you got to pack if you're going to do a long trip to Mars.
Yep.
Is if you can set up a whole bunch of these MOXIEs, create a small forest of them worth of trees making oxygen in a place where humans will be, then you can have humans on Mars breathing oxygen that's made from the Martian atmosphere, and they think producing even enough oxygen to create a fuel to get the humans back from Mars if we so desire, or if they so desire to leave.
Yeah, oxygen as a fuel is incredibly important. So that in itself, not just for oxygen, not just for breathing. It's for burning.
So all the initial testing over this past year or so have been successful.
Awesome.
Yeah.
I love MOXIE. That was one of the experiments I've been most excited about. MOXIE's Oxy generation. So fantastic. And it's just really exciting when something like this, you're like, is it going to work? It should work. It actually works. We're actually producing oxygen on another planet. This is so cool.
Yeah.
Pretty awesome there. I want to not run on the heels of the oxygen here. I want to take all the oxygen out of the room. But Blair, you were talking earlier about like anthropologists trying to figure out like what makes humans so special, what makes, and Justin, we're talking about like, oh yeah, humans, we think we've got it all. And then it turns out there's an animal out there that's doing the stuff that we thought that was just what we could do. Well, we talk all the time about humans' amazing ability to detect patterns and to look for patterns in nature. Well, we're not the only ones who look for patterns. And in fact, humans and our ability to, persevere even when sometimes there's no reason to, because we think we're going to get a reward, or we think there's some good reason underlying it, or just we have reasons for the things that we do. Yeah, well, it turns out monkeys do that too. Monkeys do that too. Yep. Researchers at Columbia University decided that they were going to dig into kind of the question of what motivates animals to do things, right? What we know that humans will very often continue to try and solve an unsolvable task, believing that there's just some kind of trick that they haven't quite learned yet. There's something that they just haven't quite gotten it, and there's a pattern there, and I'm going to see it, and I'm going to find it, and I'm going to solve this puzzle. Humans do this all the time. And what is it about our motivations to do these things, and do other species do it? Well, at Columbia University, they decided to ask monkeys if they would try to continue solving unsolvable tasks. And so they set some tasks before some monkeys, and well, these rhesus monkeys were first trained to solve puzzles that they learned through trial and error. So tried it. They made mistakes, and then they figured out the answers. And there was a learnable rule and a learnable order during the training, but then in the testing period, there was no rhyme or reason, and they even started giving rewards at weird times, and they just messed up the whole system, so that even like what the monkeys were getting their water reward for, it didn't make sense. But the monkeys kept going, and they found that there was like one of these monkeys in particular just kept going and going and going. And then they tried another monkey that they had not trained as much in this original training paradigm. So they're like, well, what about a monkey that hasn't really learned that there is a solution? Throw that monkey in there and see what they're going to do. That monkey was even more gung-ho than the original monkeys.
Never tell me the odds.
The monkeys, yeah.
Do you think this is related at all to the sunk cost fallacy? I was thinking about that.
Yes. So exactly how far you go, and if they've received some amount of water reward, maybe they're going to get more water reward. And the researchers themselves, they say, well, this was a water reward, so this is kind of survival based. Water is something that's very important to an animal. It wasn't like a special treat. Right? Where a special treat is like, ooh, this is just exciting and different and not something they get all the time. This was something that is physiologically important to them to get water. And so they wonder whether or not there would be different results if it were a different reward, or if there had been no training ahead of time, how it would have turned out. But yeah, it is an interesting question. How much effort you put in before you cut and run, right? Where you go, oh, this isn't going to go anywhere. This definitely would fit into that. But what they ended up creating somehow for these monkeys, they created an intrinsic reward task that kept them focused on just trying to solve the puzzle instead of gaming it to reach some solution. And I think that's interesting also. Yeah.
But the monkeys are getting stochastic feedback, not based on really anything that they're doing. And yet their attention is, what I'm picturing? I'm picturing that part of the casino floor where they have the digital slot machines and the zombies, the zombie gamblers that sit there and just keep.
It's not even this anymore. It's this now.
No, it's not. Yeah.
It's not as fun.
Hitting buttons just like a monkey on this behavioral task. Yeah.
It is a non-skill. The least input from the gambler sort of gambling that exists.
Yeah.
Where you don't need to think or decide, you just do it again. And maybe you get a result, maybe you don't. It has really nothing to do with your input, but you keep inputting.
But the difference is you tell the person working the slot machine that there's a way to get a surefire jackpot if you hit the right buttons in the right sequence. You can do it.
What you do is you wait for somebody who got up from a machine without hitting a jackpot, and you go sit where they were sitting, because that increases the point to which the thing's going to pay out, and that's how you win. So
but the payout, that's the sunk cost fallacy, right? So how much money you put in versus the payout. Whereas here for these monkeys, they're just spending time and they're hitting buttons and they're getting an occasional water reward. But it's a very interesting situation. So is there? The researchers say they were surprised to see that they put in random inputs and got very stable outputs out. And is this because even monkeys have some kind of a belief that there is a structure and that you can convince yourself that you're learning the structure. You take internal cues, though, as opposed to actually external cues to determining that structure. There's a pattern somewhere. Monkeys, just like people. We'll keep on hitting the buttons, pulling the levers, doing all the things. It's gotta be big payout, big money, big winner. Potentially a big win for chronic pain will also potentially
Pain wins again.
yeah, a big win for chronic pain sufferers and also for cancer sufferers. A new study published in Science Translational Medicine was looking into a molecule, a metabolite that comes out that's it's produced by sensory neurons. It's called BH4. And BH4, the more inflammation you have, BH4 gets produced by the sensory neurons, and then it drives chronic pain. So BH4 is there in the pathway, and the concentrations of it correlate with pain intensity. And so these researchers were like, let's look at BH4 and see what we can find to target it and get rid of BH4. What can we do? And so they looked at a bunch, like 1,000 different FDA medications, and they found a couple of drugs that are used to treat schizophrenia actually might have analgesic effects along this BH4 pathway, including clonidine, flufenazine, which is an antipsychotic. It blocks the BH4 pathway in injured nerves, and they were able to actually demonstrate the effects on chronic pain following nerve injury in vivo, so in a living organism. And then, so flufenazine blocked BH4, reduced chronic pain. That's awesome. But they also found a link between a lung cancer pathway, the EGFR KRAS signaling pathway. It's highly found in lung cancer, but it's involved in multiple cancers. This pathway, the EGFR and the KRAS genes are very frequently mutated in lung cancer. And they determined that this connection actually triggers BH4 or doesn't trigger BH4. So BH4 can actually be stimulated by this gene pathway in lung cancers and start to drive lung cancer and chronic pain in cancer patients themselves. And so this could lead to new treatments using already FDA approved drugs for lung cancer and also chronic pain.
Okay, wait a sec. Hang on. Which comes first? No, super important because because the way you said it sounded like somebody's got lung cancer, and that's causing this BH4 feedback loop thing to then increase inflammation and chronic pain. Because if that's the case, that chronic pain in your knee or your lower back should go get your lungs checked. It could be like an early indicator in that way.
No, not always. Yeah, not completely connected. But BH4 is because it's so connected to sensory neurons all over. It was just interesting that they found this link as well, tightly linked and correlated to these mutated cancer genes. But they did find in mice, not in humans, but they were able to show that when they decreased the amount of BH4 in mouse models with lung cancer, there were fewer tumors that grew. So it reduced tumor growth by reducing the BH4 that was being expressed. So there's a lot of...
would almost call it the other way then. Then if you got chronic pain, you better watch out because you're not at risk for lung cancer, which is like, I'm already in chronic pain. Now you're going to throw this at me? That's
not necessarily. They don't drive them entirely. But anyway, interesting connection, crosstalk between chronic pain and cancer, and potentially for both of these things, the big take home from this study is that they looked at FDA approved drugs to see how they would impact that BH4 target and pathway. And this could potentially lead to non-opioid chronic pain treatments to reduce chronic pain and potentially help with treating cancers.
Our whole body is a Rube Goldbergian machine. And we don't know which dominoes are connected. This is what I'm taking away. It's just, there's so many times that we have drugs for one thing that turn out to be good for something else. And it's because there are weird things happening in our body that we don't understand. And they're connected in ways that we don't have a handle on yet.
But we're figuring it out.
We're figuring it out. That is important.
Cool yeah.
It also is really good when you can get an FDA-approved drug approved for use on something else because it can be used very quickly.
Especially if you're a pharmaceutical company, which is the part of this that always should be a little bit of an asterisk that always makes me a little nervous when I hear about a drug that has this great benefit somewhere completely else in an unattended or off-label usage or however you want to, is because from the pharmaceutical company perspective, looking for these things is a gold mine. You already have a drug that's FDA approved to be safe in humans. You already have a manufacturing process that doesn't need to get recertified or anything like this. You already have everything you need to just make money right away by selling this product to a different group. And so I like that we can all, I understand what you're saying, Blair.
So my counter is,
because
if a drug has been around for long enough, then you can have generics. So that's the other positive thing about looking at FDA previously approved drugs is it's much closer to being away from the kind of proprietary restrictions where you can have generic versions of the drugs and you don't have to wait for that to happen in however many years.
Yeah, not necessarily, because then they can say, oh, we tweaked it to so slightly, now it's better for the... This is what happens. But to your point, though, also, because of the, what do you call it? The Rubik's Cube, the thing?
Rube Goldbergian.
Thank you. Rube Goldberg. Yeah. Thing about how the body is all interconnected. We make these amazing discoveries by having so many, thousands or a million people on a drug that there's this other effect. Mm-hmm. And why is that? And now and I love that if there's a way to do a non-opiate painkiller, then the question becomes, are opiates actually addictive or is pain relief addictive?
That seems like opiates definitely are. Yeah.
A lot of column B.
Yeah. So something that might have been studied already somewhere.
But in this, to your point about pharmaceutical funding, the funding for this particular paper was City University London, Harvard Stem Cell Institute, Canadian Institutes of Health Research, and Austrian Academy of Sciences, and the NIH, and the Zastrow Foundation, California. So it doesn't look as though they have claimed any conflict of interest or any pharma funds, but this particular technique that they used, they did a phenotypic drug screen to uncover these to uncover these connections and to be able to see what drugs could potentially modulate these pathways and the expression of these genes. Cool. Woohoo! Good times. Good times. You know what? I'm searching for a pattern here.
Yeah? What's that?
That I think we've finished our stories for the day.
We have. I am going to add, though, that Rube Goldberg went to my high school, just for fun.
What?
But then cool. Now we can be done.
Okay. That is a little bit of trivia that I had no idea. That is
not at the same time.
Fascinating.
It's a different...
That could have been interesting.
Yeah. And actually, it was a different building because they had torn down the old high school to build a new one. And technically, that was in an unincorporated area, so technically it was a different city.
All of the
But
load
really
bearing walls
amazing.
leaned up against each other, and then when they removed one of them, they all no. Okay,
No.
Anyway. Let's end the show, Kiki. Why are
Oh.
you stalling?
Oh, I'm stalled. Yeah, me stalling right now. I know. Oh, because Blair, I did need to tell you that rapamycin, which we've talked about before on the show as a potential anti-aging drug, is a drug that has been approved for other purposes previously by the FDA. Rapamycin, however, in its investigations, researchers were trying to figure out, well, do we just give a whole bunch of it to old people and then will it fix them? Well, it doesn't look that way. It looks as though giving a little bit of it to younger people might actually be beneficial. So Blair, keep your eyes out for rapamycin because
Get on
I think you're still in the window. The anti-aging window. We're going to keep...
I'm going to start
Blair.
dyeing my hair so I look like a cult.
Blair, you're still in the window, just so you
still
know.
in the window of hope.
You
Hurry
still
up,
have
guys.
time.
We
It's
just
not
closed
too
it.
late for you.
It's closing so quick.
Live while you can, Blair. Live while you can. That window closes quick.
All right, everybody. Thank you so much for joining us for this episode of This Week in Science. Woo! We made it through. My microphone didn't give up on me this show again. Let's see if we can make it next week. Thank you for joining us. And thank you to, I got to give our shout outs for the week. Fada, who's not here tonight, but who usually does our show notes and social media. I think Fada is at a conference this week and has been having a big week. So I hope he's out there enjoying himself and I hope to see him next week. But thanks so much for all that help. Gord, Arenlore, others who help keep the chat rooms clean and happy. Thank you for being here and for doing that. Identity 4, thank you for recording the show. And Rachel, thank you so much for editing. It is just wonderful to have everybody's help. I do also really need to thank our Patreon sponsors. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazard, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Ragan, Derek Schmidt, Don Munda, Stephen Albaran, Daryl Schmeishak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Vote Beto for Texas, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Denna Pearson, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, head over to twist.org and click on the Patreon link. On next week's show,
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, as well as twist.org/live.
Want to listen to us as a podcast, perhaps while you set up your 2000 domino art experiment? Just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to these stories and studies will be available on our website, www.twist.org, where you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistmania@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put twist, TWIS, in the subject line, or your email will be spam filtered into the soup that is a metamorphosis inside of a pupa.
unless it's stuck in a pocket and then it'll like come out in the wash later.
You gotta put twist in the subject line. That's how it ends up in the pocket.
Yeah.
Otherwise, it's just lost in there.
Okay.
Yeah.
If you haven't got a pocket, you can try Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there is a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.